Hello, General Biology. Welcome to our first week as a flipped classroom. This is really where it comes down to you being able to review the material and come back with questions, and then we can work through some of the harder things together. So, as a flipped classroom, it's going to be an asynchronous kind of a style where you can do this at any time during the week, but at some time you need to follow the syllabus, what we're doing, and cover the material that you're supposed to be covering. So right before spring break, we finished replication. You need to go back and look at the overall process, find a good YouTube video or two and watch them, and then find the slide that summarizes the enzymes that are involved. So you know where each of the enzymes act, excuse me, and at what point in the replication process they act. So whether it's during initiation, elongation, or termination. So cut it up into chunks. For the week of March 23rd, so that's our first week after spring break, we're going to be covering gene expression. So that's chapter 17 in Campbell's and chapter 9 in the Cell Concepts website. If you remember gene expression, I introduced the dogma of biology, the central dogma, DNA to RNA to protein. The DNA to RNA is transcription, and RNA to protein is translation. So chapter 17 has to deal with the transcription and translation processes, not necessarily replication. That's chapter 16. So go into the modules. You want to be looking at modules 6 and 7. That's way at the bottom. And you'll find the slides for chapter 17. You should probably go back and review chapter 16 as well in replication. You want to start working through that. Find the lectures, the audio taped lectures, and you can listen to the lecture as you go through the chapter 17 material. You can download those lectures to a Dropbox or listen to them through a web browser or however you want. And that's also available through the modules section in Canvas. For this week, we'll have another quiz, quiz 5, that will be on let's see if I remember correctly. It's on the cell signaling, so that's from a couple weeks ago, and also on DNA structure. So quiz 5 will be administered through Canvas. You'll have probably a 2-day open window. I'll probably make it Tuesday and Wednesday, which would be the 24th and 25th, to do that quiz online. You'll also have the Cell Concepts Chapter 8 that's due on Wednesday as well, and that's on the DNA replication section. So you want to finish Chapter 8 in GRL. You'll have a quiz, and in the meantime, you want to go over chapter 17, listen to the lecture, look at the notes, and really focus on what you want to be able to do is take a piece of DNA, so write out a gene, and take it through the transcription and translation process. See if you can do that with no special things like splicing. That's something that's more complicated. But take a piece of DNA through it. See if you can transcribe it to RNA and then translate it to protein using the genetic code that's provided in the notes. So other things that you want to focus on, gene structure. There's a slide on what genes actually look like in prokaryotes and eukaryotes. Again, for both the transcription and translation processes, take them and cut them into chunks. So cut them into an initiation stage, elongation stage, termination stage. What happens in each of those stages? What are the enzymes that are involved? For example, RNA polymerase is the enzyme that does most of the work in transcription. In translation, it's the ribosome. I basically just call it an enzyme. That's the enzyme that does all the work. What are they doing during each of those sections? You also want to be able to compare and contrast the processes in prokaryotes and eukaryotes. The gene structure and some of the modifications are different between prokaryotes and eukaryotes, and there's a slide that highlights that. So go ahead and give it a try. Leave your questions in the comments section of this video, or send me emails, as many as you want. I will respond to them pretty easily. So let me know how it goes, and good luck.